Hello and welcome to Nikolay's genetics lessons, and in this video I'm going to talk about gel electrophoresis common mistakes. This is going to be my first video in a series of videos, and if you study biology, genetics or microbiology, running gel electrophoresis would be just a part of your routine, and of course you would make mistakes. And one of the common mistakes would be to get a gel picture like this, where we have a smear. So how this can be explained? First of all, I want you to pay attention at the wells, and as you see, still a lot of material are left here. So what might cause this? Probably this is caused by aggregation of the proteins with DNA. So our DNA sample is contaminated with proteins. Also, if we would take a look at this smear, we would see that if our wells here, our DNA moves in this direction, and this smear moves faster than our bands where most of the DNA is present. So this tells us that here we have DNA which is smaller in size, so probably our sample also contaminated with DNAs, which shares our DNA, and smaller pieces are present here. So we have to include in our sample preparation certain steps in order to avoid DNAs. DNAs is protein, so our steps have to include degradation of the proteins. And another source of contamination can be RNA. So we also have to degrade RNA to get a better picture. Sometimes we also can get this type of smear if our electric current is set too high. So you have to decide which mistake you made, but basically I gave you common mistakes that leads to pictures like this one. And one more advice would be do not vortex your sample. Instead, just gently shake your test tube, because sometimes when you vortex your sample, it also can degrade your DNA material. And couple two more hints. Another variant why you can get a picture like this would be that your sample is just too old and degraded, or your technique of extraction have to include liquid nitrogen, so it would also prevent degrading. And also you have to be sure that materials that you use also fresh and not old materials. So sometimes it doesn't depend on your extraction techniques, but also you can get such picture just if you use a sample for extraction that is just too old, and such a process of the degradation of the DNA would take a place just a natural way. So use the fresh material. Such a smear also can be caused by small proteins present in a sample, and also you can get such a picture if your gel is prepared the wrong way. And how do you know it? In order to know it, you have to also load in one well a ladder, so you would see if in your ladder reference ladder of known sizes of the DNA, you would see also a smear. So probably that means that your gel is made with a mistake, so you have to check your protocol. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.